Okay, hey everyone. Today's lecture we're going to talk about fat metabolism, protein metabolism, and gluconeogenesis. We're going to start discussing a little bit about gluconeogenesis since we ended our lecture last time talking about carbohydrates and glucose. So I think the best way to start this lecture is to actually talk about how we generate glucose in our liver and then how we can use that as a fuel in periods of starvation or excess exercise, usually plus 120 minutes or so. So just a recap from last week when we were talking about an energy yield of one glucose molecule, referencing skeletal muscle and the brain, can yield about 32 to 36 ATPs. It depends on what resource you read, but for the most part, this is how I broke it down for you. Glycolysis, we can produce 2 ATPs. Then the FADH and NADH that are produced, we can form 4 and 6 ATPs, and then 2 times 12 when we send that through, that split through again, like we talked about last week, twice around the Krebs cycle. So one glucose molecule has the ability to give us about 32 to 36 ATPs, to give you a good reference point. Okay, so to recap, we have 3 different energy sources, right? Last week we talked about immediate energy sources. We started talking about non-oxidative energy sources, which was our fast glycolytic system. And then now we're going to be talking about oxidative energy sources. Now, if you remember from last week, we talked about the glucose molecule could have one of 2 fates, right? If we're exercising at a very high intensity, we could keep exercising and reduce pyruvate to lactate, correct? And then we could keep exercising. That would be non-oxidative. However, if the energy requirements of the cell says we have time to make more ATP, that pyruvate will get converted into acetyl-CoA and will go into the Krebs cycle for oxidative metabolism through the electron transport chain. Okay, when we start talking about gluconeogenesis, we have certain body stores of carbohydrates, right? Last week we talked about how we have glucose circulating in the bloodstream, as well as we have glycogen stores within our muscle fibers. To give you an idea, we do have very limited carbohydrate stores. The more you train, you can enhance your carbohydrate stores or your glycogen stores. However, just to give you a reference point, the daily requirement of the brain is about 120 grams out of the total body's need of 160 grams for the day. So our brain takes a lot of glucose for it to run. So our body fluids take about 20. We also have stored about 190 grams in our in glycogen form, and total giving us about a stored total in our body at any given point in time, 210 grams. So ultimately, the amount that is stored is adequate for only one day's need. Right. All right, so let's talk about gluconeogenesis a little bit. During periods of starvation and during exercise, the body needs to resynthesize fresh glucose, right? So for example, when we talked about last week, we briefly talked about this. So we can talk about how marathon running or some sort of triathlon or some sort of very long endurance event, this is where individuals start to kick up gluconeogenesis. Major site for this process is in the liver. It does occur in the cortex of our kidney, but the amount of glucose produced is 1/10 of that made in the liver. So again, this can occur in the cortex of our kidney. However, the major source of gluconeogenesis where it's being generated is within our liver. Okay? I want you to all know that. Right. So non-carbohydrate precursors enter this pathway and are used to regenerate glucose. So non-carbohydrate, again, meaning things like pyruvate and lactate. So if you can see down here, we have pyruvate down here in our cytosol, and we can bring it back into the cell. This is all happening within the liver. And this is our pathway, ultimately, for the most part, that comes up to this point, and then it finishes up the pathway to make glucose. And then we can also start down here. You can see how if we start with lactate, it'll eventually be reduced to pyruvate, go back up the chain, back out into the cytosol, and we'll go back up the chain making fresh glucose. So ultimately, it's non-carbohydrate precursors that enter this pathway that can produce fresh glucose. So I said last week that sometimes lactate actually isn't the bad player. We'll talk about this a little bit more in the class. where a lot of individuals say, oh, we have excess muscle soreness because of the lactate buildup. That's not at all true. We actually use lactate for a lot of other things. We can use it in shuttle systems and bring it to reduce it to NADH and FADH2 to shove it through the electron transport chain to produce more ATP. And we can also use lactate here in this Cori cycle where we can reproduce new glucose. So lactate isn't the bad person that we always grew up as athletes, for the most of us, learning that muscle soreness was due to lactic acid buildup. So we'll talk how that's not true later on in class as well. Okay, for gluconeogenesis, this metabolic pathway, the energy requirement is 6 high energy phosphate bonds are used, so 4 ATP and 2 GTP, and then 2 molecules of NADH. So ultimately, we do need to put some energy into this cell in order for us to produce a glucose molecule, which then that glucose molecule can go back out in the bloodstream and we can use it for anaerobic or aerobic metabolism, right? So we could either get a net gain of 2 ATPs from that, or if it was an anaerobic task, if it was an aerobic task, we could get something like 36 ATPs from it. So even though we're sacrificing a little energy, we could ultimately get between 32 and 36 molecules of energy from producing this molecule of glucose. Okay, so gluconeogenesis, this pathway is affected by rate. So the rate of gluconeogenesis is affected by substrate availability. So how much lactate and pyruvate do we have in order for us to use to produce this glucose? Allosteric effectors and hormones such as glucagon. Remember, if we talked about last week that an allosteric regulation of an enzyme this is when a binding of a substrate can alter the shape and function of that enzyme, right? So it can be inhibitory or stimulatory. Okay, glucagon. What is glucagon? This is produced by our alpha cells in our pancreas. This works to raise our glucose and fat levels in the bloodstream. So this works the opposite of insulin. So where insulin and glucose, where insulin takes glucose out of the blood, glucagon tries to put glucose back into the blood. So these two, glucose and insulin, work as a negative feedback system to each other. Okay, so that hopefully you understand that concept as well. Okay, so here in our pancreas we produce insulin as well as these alpha cells, right? So low blood glucose levels, glucagon is going to raise in our cell, and glucagon will also stimulate our liver to produce more glucose because we need to have it put into the bloodstream. It's gotten too low. Now the difference is, with insulin senses the fact that we have too much glucose entering the bloodstream, so insulin needs to go in and tries to get some of that glucose from the bloodstream into our cells. Okay, so gluconeogenesis is stimulated by high levels of lactate, glycerol, and amino acids. So ultimately what this means is, again, we're at periods of starvation or we have exercised and used a lot of our energy stores up, and high levels of these indicate that. So these will indicate to our cells that, okay, we need to kick it up a notch and produce more glucose. So this will stimulate gluconeogenesis in our liver. All right, so here's the pathway for the most part, okay? So if you look over here, this big square is our liver. This is our muscle tissue, and this is our blood. So we're traveling in the blood right here. So ultimately, we're exercising a lot, or we're in periods of starvation, and let's just say we're exercising and we're producing a good amount of pyruvate and lactate with our exercise. We can use this lactate, can be shuttled out, sent to the liver, and we can use it as a precursor in gluconeogenesis to follow up the pathway to now make fresh glucose. Our liver has made this new molecule of glucose. We can shove it back out into the bloodstream, and then our muscles can use it once again for energy, to produce energy for muscular contractions to keep occurring. So this is what's called the Cori cycle. So this is where gluconeogenesis takes place, right? We said in the liver. However, this whole cycle where we can produce pyruvate and lactate, and instead of them being metabolic waste, which usually they are, we can actually still use them with gluconeogenesis producing glucose. However, this is what's called the Cori cycle. Okay, so the lactate that enters the liver is oxidized into pyruvate because of the low NADH-NAD ratio in the liver, right? So remember we talked about those ratios and how they pretty much govern all of our metabolic reactions. So a low NADH to NAD ratio will stimulate this. Pyruvate is then converted to glucose. Alanine also is formed from pyruvate by a transamination and exported from the muscle to the liver, right? So if we're ultimately breaking down protein, which we'll talk about in a little bit, it can also be a precursor in this Cori cycle. Okay. So moving on, we're going to actually talk about lipids now. So we're out of carbohydrates, we're out of glucose, we're out of all of that. Now we're completely to a totally different store, which is our triglycerides, and we're going to be talking about lipid metabolism for the next few minutes. Okay, so when we are talking about lipid metabolism, we are going to be mainly breaking down triglycerides. Okay, so they usually are abbreviated if you read in your text TAG, T-A-G, and you may think to yourself, there's no A in there, why is a triglyceride named as that? Well, actually their technical name was triacylglycerol, and there was an A in there, so in case you're wondering why TAG is abbreviated that way, the official name is triacylglycerol. But what are triglycerides made from, right? So 1 glycerol and 3 fatty acids. So right down here we can see 1 glycerol and 3 fatty acid is a triglyceride. We will break these down to attain energy to perform metabolic work while we exercise. And this is usually during low intensity to moderate intensity exercise. We will mainly be accessing our fat stores for energy. And like I said last week, that's because mainly one molecule of fat has the potential to give us upwards of 125 to 130 molecules of ATP we can use for energy, as compared to a glucose where that can give us the max amount of energy is about 36 ATPs. So if we use fat, we're a little bit more efficient in that sense. Okay, so the purpose of lipid metabolism and beta oxidation are to produce acetyl-CoA and high energy reducing equivalents, which are NADH and FADH2. So the whole point of breaking down a fat molecule is to ultimately get NADH, FADH2, where we can shuttle that again to the electron transport chain to go through oxidative metabolism and give us that ATP. Okay, right, so you can see with this diagram right here, if we use glycogen or glucose, but right here this diagram is saying glycogen, we can break it down into glucose to pyruvate. If we are going aerobic, so we want to go oxidative metabolism, we can break it down. Instead of the pyruvate going to lactate to keep this shuttle going, we're going to break it down into acetyl-CoA. Acetyl-CoA can then enter the TCA cycle, which again is the same thing as the Krebs cycle. And then we make all those high energy reducing equivalents and we send it to the electron transport chain, and here's where we get our ATP. Now, similar pathway over here, if we use a triglyceride or a phospholipid, ultimately we're going to go through this lipolysis metabolic pathway. I'm going to break down the fatty acids, and we're going to ultimately, at the end of beta oxidation, is to produce acetyl-CoA, which again, it's the same thing as if we were breaking down glucose and we were going to go to aerobic metabolism. We're also going to break this down. The end result is acetyl-CoA. Now all these molecules of acetyl-CoA can enter the Krebs cycle and go through the same process. And ultimately, ATP synthesis. Okay, so last slide of this video, we are going to talk about where triglycerides are stored. They are stored in 3 places. The adipocytes, right, so our fat cells, within our liver, and also within our muscle. So some are stored between our muscle fibers, while some are also stored within the muscle. So intermuscular fat stores. The ones that are within the muscle fibers themselves technically usually are adjacent to the mitochondria. That's just a fact for you to know. But the main 3 storage sites I want you to know we can access from: adipocytes, liver, and our muscle. That's where they usually like to be stored. Okay, so how we transport fat and blood lipid levels, what they depend on, will be in the next video coming up.